Uh, Hi, good afternoon, and welcome to the Cancer Lightning Talk session. My name is Jyoti Menon, and I'm from the University of Rhode Island. I'll be moderating the session today. So, as all the presenters are aware, we have 3 minutes to give a brief overview of your research. And so, our first presenter is Camilo Osorio from Brown University. So, go ahead, Camilo, when you're ready.
Hello everyone. My name is Camilo Osorio. I'm a postdoctoral research fellow at Brown University. Today I'm going to be talking about how SHIP2 regulates gastrointestinal epithelium development by modulating the biology of Ostrich-positive crypt stem cells. A little bit on the background, SHIP2 is a protein tyrosine phosphatase encoded by the PTPN gene. It is a ubiquitously expressed protein essential for the development and maintenance of multiple tissues and organs. However, whether and how SHIP2 regulates osteoblast development and function remains unknown. Osterix is a zinc finger containing transcription factor essential for osteoblast differentiation and function. Osterix is reported to be specifically expressed in osteogenic cell lineages. In this study, we seek to explore the role of osterix positive cells, which we presume would be in this we seek to explore SHIP2 role in osterix positive cells, which we presume would be specific to bone-forming osteoblasts. Next slide, please. To explore the role of SHIP2 in mouse skeletal development, we generated mice in which SHIP2 was inactivated in osterix-positive expressing osteoblasts. Mice were born at the expected Mendelian ratios and appeared normal at birth. However, their growth and weight gain were dramatically impaired when compared to the controls. Unexpectedly, pups began to die at 2 weeks postpartum. Necropsies performed revealed gross gaseous distension and hemorrhage along the length of the small and large intestine. Histopathological examination of the gut samples revealed dramatically truncated villi in the ileum, but no apparent evidence of inflammatory infiltrates or tissue necrosis. To understand how SHIP2 in the ostrich-positive cells causes GI pathology, we carried out live cell lineage tracing studies. Immunostaining with antibody against murine ostrich localized mainly to the base of the crypts, which are predominantly populated by stem cells and Paneth cells. Thus, our results strongly suggest that ostrich promoter is active and ostrich protein is expressed in the crypt-based columnar stem cells. Staining of ileum frozen sections revealed fewer EDU-positive cells in the crypts of SHIP2 knockout mouse when compared to controls. Similar dense populations of normal-appearing enterocytes and goblet cells were seen in both strains of mice. This data suggests that SHIP2 is critical for self-renewal and proliferation of ostrich-positive crypt stem cells, but has minimal impact on their differentiation. Next slide. SHIP2 was associated with compromised ERK activation and enhanced SMAD1, 5, and 8 activation, but no apparent effect on caspase-3 cleavage. This data thus suggests that SHIP2 is required for VMP-evoked signaling in the gastrointestinal stem cells. Tissue from 76 colon cancer patients were retrieved and stained, and 92% of the cancer samples exhibited strong ostrich signaling. In contrast, only 20% of normal tissue sample express ostrich-positive tissue. This data confirms that ostrich is expressed in the GI epithelium, and its expression is substantially expressed in GI neoplastic tissue. To take away, the GI tract expresses ostrich, which is novel and has never been expressed before, and future work of inquiry are to further explore the ostrich expression and its potential therapeutic, diagnostic, and prognostic values in GI malignancy. Thank you very much.
Excellent. Thank you, Camilo. Our next presenter is Marcel Actonhagen from University of Delaware. So, Marcel, whenever you're ready, please go ahead and get started.
Thank you very much for the kind introduction. So my name is Marcel Achtenhagen. I'm a fifth-year graduate student from the University of Delaware, and I'm currently working in the chain laboratories. And I'm going to be talking to you today about the total synthesis of Sigidal-A, which is a selective inhibitor for the hepatocellular carcinoma. So just as a little bit of background here, in 2016 it was about 40,000 new cases and 27,000 new deaths. So these are quite some eye-opening numbers that we need to address. So our goal with this project is to complete the total synthesis of Sigidal-A and to also provide a more selective inhibitor in the HepG2 cancer cell line with respect to doxorubicin. So kind of the current drawbacks of doxorubicin include myriad health side effects, which are not limited to acute cardiotoxicity. So that's something we need to address. And the other issue that we have is the IC50 in the HepG2 cancer cell line, which is 5 micromolar. For Sigrid-LA, the HepG2 IC50 is 61 nanomolar, so we are orders of magnitude more potent and selective with that natural product. Next slide, please. In terms of the chemistry, there's a lot of organic chemistry going on here, and the takeaway message is that we can take commercially available +3 carrying, which is about 3 cents a gram, and within 15 steps, we can get to our first key step. And this is actually really some exciting chemistry that we're doing here, as well as some successful chemistry. So we have shown that with our TMS enolate 4 and our double benzoyl chloride 8, we can do our first key step. So that's an orthoquinone enolate methide coupling to generate the bottom half of the natural product. So we're essentially 1 key step away from completing the total synthesis. Next key slide, please. In terms of our endgame strategy, we had kind of suggested an oxypane ring formation here. And what we're going to do here is get to the diene that is shown in the middle of the screen there, and then do a selective hydrogenation of the disubstituted alkene, followed by a deprotection. And once we have that, this actually sets us up for a mercurinium intermediate that will allow us to capture the free phenol, generating for us the 7-membered oxypane ring, thus building the core structure of the natural product. And from there, it's about 2 steps further to get to the final end product. But again, we would have a successful completion of that, which would be able to displace doxorubicin from there, providing a much safer alternative than doxorubicin. So with that, I would like to, again, thank everyone for listening. I would like to thank Professor William Chain for allowing me to work in his laboratory, as well as the University of Delaware and the funding partners listed below. Thank you.
Thank you, Marcel, for that excellent presentation. So if the audience has any questions, please feel free to add those questions on the screen, and we'll address them in the end of the session. Our next presenter is Brendan Riley from the University of New Hampshire. So Brendan, whenever you're ready, please go ahead and get started.
Thank you very much. My name is Brendan Riley. I'm from the University of New Hampshire, and I work in Dr. Sarah Walker's lab. I'm going to talk to you today about repurposing drugs to target STAT3 in cancer. So our protein of interest is STAT3. It's a transcription factor that becomes phosphorylated, enters the nucleus, and affects transcription of genes that are involved with things like metastasis. So to find a drug that would target STAT3, we used a publicly available tool through the Broad Institute called CLUE. And CLUE uses L1000 gene expression signatures. And what you can do is take a list of genes that are up or down regulated from some kind of effect. In our case, it was knocking down STAT3. You can take that list, put it into CLUE. It will compare it to other gene signatures from other perturbagens and give you back a ranked list of perturbagens. These things can be stuff like drugs, chemicals, knockdown experiments, overexpression experiments. And what we were interested in was drugs to see that would kill cancer cells. Next slide, please. So initially what we found were 4 statin drugs, lovastatin, simvastatin, atorvastatin, and mevastatin, that CLUE indicated would have a similar effect to the knockdown of STAT3. So what we first wanted to, So what we first wanted to do was look at how that would affect cell viability. We do this by growing our cells up in a 3D spheres, and then exposing them to the drugs for 7 days, and then putting cell titer glow on, which measures the amount of ATP available in that cell well. Looking here at our preliminary data, we can see that the 2 cancer cell lines, HEA8 and OVCAR8, showed lower cell viability when compared to an immortalized LP9 cell line. We next wanted to look at how the mechanism of action would work on this. So we took our 2 ovarian cancer line cells, we knocked down HMGCR, and looked at if it changed the total amount of STAT3 expression or the total amount of phosphorylated serine STAT3. Our preliminary data indicates that it did downregulate both of those. Next slide, please. Why is this important? STAT3 is a good target to create more targeted cancer therapies. Drug repurposing could be important in shortening both the time and the amount of money it takes for new cancer cell therapies. Statins have shown to have protective effects in ovarian cancer cell patients. Our future work is just looking at how this mechanism may occur. One suggested one is this figure down here by Arnold et al. Last, I just want to thank INBRE, Dr. Sarah Walker, and my lab mates for their help in my research. Thank you.
Thank you, Brendan. The next talk is by Kanayo Ike from the University of Vermont. So Kanaya, whenever you're ready.
So the goal of my master's is to determine the transcription factor GLI2's role in B cells, specifically in class switch recombination, CSR. CSR is when a B cell makes the change from expressing IgM antibody to another isotype. This occurs by creating a new germline transcript at the DNA level. Different antibodies mediate different immune responses. For example, IgE antibody is selective for responding to allergies and parasites, and IgA is found commonly at mucosal surfaces. In the rare cancer Waldenstrom macroglobulinemia, also known as WM, B cells have an impaired ability to undergo class switching despite normal CSR machinery. Our lab has shown a role for GLI2 in regulating IgM secretion in WM via regulation of IL-6 receptor. GLI2 is a zinc finger transcription factor, and we have found multiple potential GLI binding sites represented by the lines in between the variable region and the constant region of the heavy chain. This region is important for heavy chain expression and class switching. Next, please. Although we know that GLI2 regulates IgM secretion in WM, we want to determine the effect in normal B cells. So we generated a conditional knockout mouse model where GLI2 was deleted in B cells. Mice with this mutation have no significant change in survival or B cell development. We found that in vitro stimulation of splenocytes, the IgM levels are similar, but the IgG levels are significantly higher in these knockouts. Using purified B cells from spleens, we performed a CSR assay analyzed by surface level flow that showed specifically IgG1 is higher, but there is no noticeable change in IgG3 or IgA. More mice will be analyzed to determine significance. Next slide, please. Because CSR is important in WM, we have analyzed a few cell lines to determine GLI2's role in these malignant cells. With GLI inhibitor GANT61, we have found an increase in IgG2 and IgG4 expression. In the presence of GANT61 analyzed through qPCR, there is also an increase in the proteins related to CSR, such as AID, ANRAG1, and RAG2. This is imperative because it shows that GLI may modulate class switching. From here, we will look at the mechanism that regulates CSR and determine if GLI2 influences proliferation or survival of the mouse B cells. Thank you.
Thank you very much, Laura. Our next speaker is Cole Davidson from the University of Vermont. So Cole, whenever you're ready.
Hi, everyone. Today I am talking about a new drug target in anaplastic thyroid cancer. Thyroid cancer is the most common endocrine cancer and is usually easily treatable. However, the most aggressive form, known as anaplastic thyroid cancer, or ATC, has a median survival of only 6 months, making it one of the most lethal tumors. The mainline therapy, sorafenib, can extend patient survival rates to 11 months, but that is still an abysmal outcome. There is clearly a need for more effective, targeted therapies for improving ATC patient survival. One cellular process that has not been investigated in ATC is glycogen metabolism. Some cancer cells are known to store glucose as an emergency carbon cache in the form of glycogen using glycogen synthase, GYS1. This stored glucose can be broken back down by glycogen phosphorylase liver or brain isoforms to produce energy and make new building blocks for replicating cells. Our straightforward but provocative objective was to determine if ATC cells metabolize glycogen and if this represents a novel drug target. Next slide. We use thyroid samples from human patients to measure glycogen enzyme expression. As we can see here, GYS1 and liver phosphorylase levels do not differ greatly between the types of thyroid tissue. However, normal thyroid tissue does not appear to express brain phosphorylase at all, while even benign thyroid adenomas up to ATC samples significantly upregulate the brain isozyme. We therefore profiled 7 cell lines, including normal thyroid cells, the less aggressive papillary and follicular thyroid cancer cells, and of course, HEC cells. Using qPCR, we saw again higher levels of the brain form of phosphorylase and the more aggressive thyroid cancer cells. We then needed to measure actual glycogen in thyroid cells. Using an absorbance-based assay, we see variable levels of glycogen reflective of genetic backgrounds across the cell lines. We can literally see the glycogen stores of various morphology in both normal thyroid cells and HEC cells using electron microscopy. Next slide. We then targeted glycogen phosphorylase with the small molecule CP91149. This caused a buildup of glycogen both in our normal thyroid cell line and in HEC cells. This resulted in reduced HEC cell numbers, but this did not dramatically impact normal thyroid cells. When used in combination with sorafenib, we saw remarkable drug synergy to dramatically kill HEC cells in vitro. Hopefully, combining sorafenib with CP will yield promising translational results in vivo. In fact, we are injecting HEC cells into mice in just 6 days to begin CP and sorafenib treatments for the first time. Perhaps looking at outside-of-the-box drug targets, such as glycogen, could one day help effectively treat patients with the most lethal cancer. I want to thank my excellent mentors for their guidance, our funding sources, as well as the audience for listening. And thank you to our wonderful host for this opportunity. Thanks.
Thank you, Cole. Really interesting work. Our next speaker is Mona Karbalawan from the University of New Hampshire. So Mona, whenever you're ready, please go ahead and get started.
Hello, everyone. My name is Mona Karbalawan, PhD candidate at the University of New Hampshire, and I'm going to talk about the role of MLR1 in Waldenstrom or WM. WM is an indolent B-cell lymphoma characterized by overproduction of a monoclonal IgM. Genome-wide sequencing identified an oncogenic mutation in mediated gene that is prevalent in more than 90% of WM cases. This mutation drives TLR signaling, leading to activation of NF-kappa B cascade. We found that MLR1 and its cofactor menin are upregulated in WM patients compared to healthy controls. So we hypothesized that MLR1 modulates the genes playing an important role in WM biology, and therefore represent a suitable target for this disease. Next slide, please. To test this hypothesis, we knocked down MLR1 using shRNA or inhibitor MI2 in WM cells and found a significant reduction in IgM expression and secretion. So our next question was how? Expression of IgM relies on regulation of 7 kilobase area between J and CMU region. So we designed overlapping primers for this area. Using CHIP PCR assay, we identify several regions with H3K for trimethylation marker binding. Moreover, we perform CHIP qPCR assay using 5 primers for 5 MLR1 binding sites after LPS stimulation to activate TLR-mediated pathway in CD19 positive B cells from healthy donors and found significant increase in H3K4 trimethylation binding at 4 MLR1 binding sites. Next slide, please. We also found that after MLR1 inhibition, H3K4 trimethylation binding at binding site 1 and 4 was reduced in WM cells. So we also tested the impact of MI2 treatment in vivo. We injected WM cells in SCID mice, monitored for tumor development, and once tumor appeared, we treated with MI2 or vehicle for 4 weeks. We found that MLR1 inhibition does not affect tumor volume, but reduced IgM secretion level in vivo, which is consistent with our in vitro data. So for future, we're interested to study the role of MLR1 in WM more globally using RNA-seq and CHIP-seq studies. Thank you for your attention to my presentation.
Excellent. Thank you, Mona. So please remember, if you have any questions, you can write them down using the Ask Your Question area on your screen so that we can have our speakers address them at the end of the session. Our next speaker is Lisbeth Avalos-Irving from Rhode Island College. Lisbeth, whenever you're ready.
Hi, I'm here with
my colleague, George Victorino. So I'm Liz Smith. I'm a research assistant and lab manager at Rhode Island College.
And I'm George. I'm a postdoc at Rhode Island College.
And in our lab, we work with DNA polymerase theta, which is an error-prone DNA repair enzyme associated with microhomology-mediated end joining. More recently, a study by Junhoon Yoon implicated Pol theta in translation synthesis opposite UV lesions, indicating that error-prone translation synthesis by Pol theta actually protects against skin cancer. In collaboration with Yale, we've identified the sun-exposed melanoma-derived Pol theta variant, which we hypothesize may have reduced DNA repair function based on its location within the active site. To test this hypothesis, we can characterize each step of the biochemical pathway shown in Figure 2. Next slide, please.
We are going to look at the highlighted part of the mechanism. To test our polymerization of our variant, our lab did a biphasic burst assay, which focuses on the polymerization and release of product. What we expect to see in a burst assay is a biphasic trend where the initial part of the graph is the burst of product formation and then followed by the linear rate-limiting product release. The assay was run by pre-mixing the DNA substrate shown on the right side of the slide with Pol theta and then mixing it with the correct nucleotide over multiple time points, leading up to 0.6 seconds using a rapid quench flow. Using gel electrophoresis, we visualized and quantified product formation. When quantified, we saw that the L variant has an observed polymerization rate that is 12 times slower than the wild type, and its steady state rate does not fit within our parameters of error that we expect for a biphasic burst, suggesting altered activity.
Next slide,
Next
please.
slide.
In collaboration with the Delaney lab at Brown University, we generated DNA with the cyclobutane pyrimidine dimer, or CPD, which is known to disrupt replication. As stated earlier, Pol theta is capable of translation synthesis, and it does this by adding a nucleotide opposite the UV lesion and UV-induced lesion and extending it in order to continue replication. Previous literature has shown mixed results about whether or not wild type can bypass CPD damage. However, enzyme conditions may have had a role to play in this. On the left-hand panel for wild type and the variant, both CPD damaged and undamaged DNA were titrated in increasing concentrations to mimic steady-state conditions. We found that when we were closer to a 1-to-1 DNA to theta ratio, wild type highlighted at the top achieved almost full extension post-bypass, whereas the variant on the bottom did not, it stalled. At higher DNA concentrations, no bypass was observed. On the right top panel, we have the ratios flipped to mimic single turnover conditions. And here theta was titrated in increasing concentrations, and we found that again at the 1-to-1 ratio, the variant stalled.
Taking these two pieces of the puzzle, we see that L2538R variant shows that altered repair pathway and completely understanding its altered mechanisms can help us identify potential biomarkers or develop potential therapeutics in the future.
We'd like to thank our collaborators, our PI, and RI-INBRE for funding this research. Thank you.
Really interesting work. Thank you. Our next speaker is Morgan Andrews from Rhode Island College. So Morgan, when you're ready, please go ahead.
Thank you. I'm an undergraduate student, and I would first like to thank Rhode Island INBRE for funding this project. So human DNA polymerase theta is a low-fidelity DNA repair enzyme. However, it has been shown to protect cells against UV radiation. The mechanism by which Pol theta selects nucleotides for nucleotide incorporation is critical for understanding genome stability. By studying the mechanism of Pol theta, further insights into diseases like cancer and mutagenesis may be revealed and could ultimately lead to innovative drug-targeting therapeutics. We tried to study this mechanism before using FRET techniques by labeling the polymerase itself. Figure 1 shows Pol theta's structure, which contains a DNA-binding thumb domain, a nucleotide-binding fingers domain, and an active site-containing palm domain. Fidelity of the polymerase is determined by Pol theta's mechanism of nucleotide selection and incorporation as the fingers domain moves in a closing manner, aligning the nucleotides with the DNA substrate within the active site. Our previous FRET study sought to site-specifically label the fingers domain of Pol theta, highlighted in purple, using transglutamase as described in Figure 2. As a reminder, Figure 3 shows how a typical FRET system works. And in Figure 4, we see our targeted sequence in purple, which is also highlighted in Figure 1. However, this was ultimately unsuccessful as there are multiple potential labeling sites, as highlighted in blue in Figure 4, resulting in non-specifically labeled protein. Next slide, please. Thank you. Thus, we pivoted to labeling the DNA substrate itself instead by using 2 aminopurine fluorescents. 2AP is a fluorescent reporter molecule and a base analog. This technique has been used to study other DNA polymerases before. 2AP is a useful probe for conformational changes since its fluorescence is very sensitive to its environment, being reduced mainly by pi stacking interactions with adjacent bases, especially during correct nucleotide incorporation. Figure 5 shows the crystal structure of Pol theta. We have identified a cancer-derived variant, L2538R, in the active site of the polymerase. We'll be comparing wild type versus the cancer-derived variant to determine if the mutant exhibits altered dynamic activity. Figure 6 shows our extendable deoxy-DNA substrates with 2AP modifications used at 2 different locations, either right at the templating base, which we call +0, or slightly away, which we call +1. In total, we use 4 different DNA substrates, as we also use non-extendable dideoxy-terminated primers not depicted in Figure 6. For fluorescence emission assays, we look at both correct or incorrect nucleotides. Next slide, please. Figure 7 shows our fluorescence emission spectra. Solid lines indicate wild type, dotted lines indicate our variant. In the interest of time, we'll be zooming in on the top left panel, which most interestingly, we see wild type with deoxy DNA behaves as expected based on other DNA polymerases, where we see a decrease in fluorescence due to pi stacking interactions during nucleotide, when a nucleotide comes in. Although the variant interestingly shows an increase in signal. And we see a similar pattern with the dideoxy DNA substrate in the top right panel, and additionally at the +1 position shown in both panels below when the probe is away from the active site. Not much difference is observed between the wild type and the variant. So in total, this suggests there's a change in the active site's environment. In summary, L exhibits different conformational dynamic activity compared to wild type. Our future works will include stop flow fluorescence studies to examine these dynamics in real time. Ultimately, we have a workable fluorescent reporter assay to monitor the conformational dynamics of Pol theta and its variant, which has never been done before. Thank you.
Excellent. Thank you, Morgan. So we'll move on to our next speaker, Julia Apitz from St. Anselm College. So Julia, whenever you're ready, we're excited to hear about your work.
Thank you. Hi, everyone. My name is Julia Apitz, and I am an undergraduate student at St. Anselm College in Manchester, New Hampshire, working with Dr. Jennifer Pace, researching FA-NA hybrid analogs as inhibitors of the Hippo pathway. So for some background, the Hippo pathway plays a role in cell proliferation, differentiation, and apoptosis. The pathway also regulates 2 transcription factors, YES-associated protein, also known as YAP, and transcriptional coactivator with PDZ binding motif, known as TAZ. And dysregulation of the Hippo pathway can lead to uncontrolled cell proliferation and tumorigenesis, making the Hippo pathway a target for cancer treatment. So as shown on the figure on the right, figure 1, there is a complex within the Hippo pathway called the YAP-TEAD complex, and that is what exhibits carcinogenic activity. So by targeting this downstream YAP-TEAD complex, we would diminish potential interferences and side effects. Next slide, please. So it has been found, searching the literature, that flufenic acid and niflumic acid are found to be potent hits in inhibiting the YAP-TEAD complex by binding into the central binding pocket of TEAD. Flufenic acid and niflumic acid are members of the flufenamides, or NSAIDs, which treat inflammation by binding to cyclooxygenase. So I use these as the target compounds for my FA-NA hybrid analogs. So my proposed hybrid analogs contain an amide bond between rings A and B, and the carboxylic acid moiety off of ring B was removed in order to get rid of any off-target side effects from binding with cyclooxygenase. These hybrids are currently being synthesized in the lab using a one-step amide coupling reaction, and some of the analogs have been computationally docked to predict their binding affinities with TEAD. Next slide, please. So the FA-NA analogs that had been computationally docked showed binding affinities that were similar to just flufenamic acid alone. This summer, 7 analogs were synthesized and purified in the lab with promising yields, as shown in Table 1 on the right. And all of these products were analyzed using proton NMR, carbon NMR, IR, and GCMS. And future direction involves confirming this purity using HPLC. And the inhibitory activity of the hybrid analogs will be analyzed utilizing gene expression of breast cancer cell lines. I would like to thank New Hampshire INBRE as well as the St. Anselm College Chemistry Department for supporting me in my research. Thank you.
Excellent. Thank you, Julia. So we have reached our last presentation, and I should thank all our speakers for staying within the 3-minute time frame that we had for each talk. So our last presenter is Zoe Chen from Dartmouth College. So Zoe, whenever you're ready.
Yeah. Hi, everyone. I'm Zoe, and I'm an undergrad at Dartmouth College working at the Moen Lab. And today I'll be presenting my project on predicting node-negative non-small cell lung cancer outcomes through survival modeling. This project matters because NSCLC accounts for 17% of lung cancer cases, which is one of the most common cancers in the US, and a fifth of NSCLC patients experience disease recurrence. Early stage patients are faced with the question of whether or not to get chemotherapy, and it's not a light decision due to the highly toxic nature of treatment. Accurate risk assessment is thus important for best decision-making practices. For high-risk patients, chemotherapy can be vastly beneficial, and low-risk patients, meanwhile, should be spared. The aim of this study is to combine socio-demographic and clinical covariates to best predict risk of disease recurrence to better inform precision medicine strategies. To do so, I investigated de-identified health records of 6,020 NSCLC patients through cancer-linked discovery. Next slide, please. Patients in our cohort tended to be older, slightly more female, and white. The two outcome variables that I was interested in were 2-year all-cause mortality, which looked at survival within the 2-year study window based on difference in dates of death and diagnosis, as well as 2-year disease recurrence, which looked at the time until recurrence or progression within the 2-year window. And this was based on difference in dates of assessment and diagnosis. Patients lost to follow-up within the 2-year window were censored. Patient similarity networks were assembled for patients with similar comorbidities upon diagnosis based on risk of adverse outcomes. For survival analysis, I used Cox proportional hazards modeling. Next slide, please. We also used community detection to identify subgroups of patients with similar comorbidities visualized in the top left, and their network community was used as a predictor in the Cox proportional hazards models. A hazard ratio greater than 1 indicated a positive association between covariate and hazard probability, whereas a hazard ratio of 1 indicated no effect, and less than 1 correlated to a reduction in the hazard. Forest plots of the survival models show that membership in the Black racial group and the younger age groups reduced the mortality hazard. Males, meanwhile, had a higher hazard for mortality and disease recurrence. Patients in Network Community 2, which included patients with chronic obstructive pulmonary disease, and Network Community 4, which included patients with diabetes, had an increased hazard for mortality, but a decreased risk of disease recurrence. These results suggest that management of comorbidities may be a challenge for improving survival. For the future, additional predictors and covariates could be evaluated to provide more personalized risk assessment for early stage NSCLC patients. Thank you so much for your time.
Thank you, Zoe. Really nice talk. So we do have one last presentation. One of our speakers has come back in. So I'd like to introduce Kanayo Ike from University of Vermont. And Kanayo, whenever you're ready.
Thank you very much. Hello, everyone. My name is Kanayo. Today, I'll be telling you about an interesting component of my thesis work that shows that inhibiting the translation synthesis pathway may not be a good strategy to improve cancer radiotherapy outcome. Generally, the success of chemo and radiotherapy stems from the ability to damage DNA in such a way that DNA replication is stalled and cancerous cells ultimately die. Translation synthesis polymerases are able to replicate over DNA damage and therefore can rescue the stalled replication fork, though they do so in an error-prone manner. So the cancers are able to come back, and they come back with even more mutations. One of these polymerases, Rev1, acts as a scaffold and recruits other polymerases to the sites of the DNA damage through its C-terminal domain. This makes Rev1 an attractive target to improve therapy outcomes. Next slide, please. So far, we have a couple of drugs that target the C-terminal domain of Rev1, and studies have shown that use of these drugs to inhibit Rev1 sensitize cells to treatment with cisplatin. In my lab, we therefore hypothesized that inhibiting Rev1 will be able to sensitize cells to radiation. Interestingly, our data presented on this slide suggested the opposite to be true. We tested different cell lines, different Rev1 inhibitors, and different doses of radiation, and we showed that Rev1 inhibition does not sensitize cells to radiation. This was interesting. One will wonder why this was the case. Well, it turned out that Rev1 inhibition triggered autophagy. Next slide, please. As shown on the left panel on this slide, autophagy was detected by immunofluorescence, as indicated by the green signal in the drug-treated cells and Rev1 knockout cells, and we use chloroquine as a positive control. We further confirmed the induction of autophagy by Western blots, and we showed that the LC3A and B and P62, which are proteins in the autophagy pathway, are more expressed in Rev1 knockout cells and drug-treated cells. Right now, we're trying to address our hypothesis that inhibiting autophagy will lead to sensitization of cells to radiation in the absence of Rev1 function, and have shown this sensitization at 5 nanomolar of bacillomycin A1, which is an autophagy inhibitor. This work is currently under review in the journal Cancers. I would like to acknowledge the members of the Chatterjee Lab at University of Vermont for their contributions to this work. Thank you very much.
Thank you, Kanayo. Really interesting talk. And thank you to all our speakers for this session. So we now move to the questions that might be there.
All right, so this is Brett Pellock from Rhode Island INBRE. There are no questions appeared in chat, but I do have one question for Laura Kehoe, if you're still in.
Hi.
Hi. So first off, thanks for being resilient and winning on the road with our audio problems earlier. The question I had for you is actually it's a more general immunology question, but it interfaces with the GLI2 function that you were saying, can change and change the classes of antibodies that are being produced by a B cell. So the general question I have is the following. My recollection is that depending on which constant region you make, that determines class switching. So if you're saying, hey, I want to be IgA or IgM or IgG, that how the antibodies are linked together or are not and are monomers is determined by which constant you're expressing. Is that true? Is it like
Yes. Yeah. So it happens at the DNA level, which is pretty unique. It is segmented like IgM, IgD, and then the DNA gets looped and cut out so that the variable region is then adjacent to the appropriate constant region. Did that answer?
Okay, yes, it does. No, I'm not an immunologist, so I was trying to figure out in my head what I remembered about class switching, and I thought I would ask you. Thank you very much.
Yes, of course. Thank you.
And I'm all set for questions, Jyoti.
Okay, so if there are no other questions, I think that will be the end of the session. Again, thank you for all the speakers and for the really wonderful work that you're all doing. I think we'll take a break now until the next session. Thank you.